Hello everyone. Now let's revise on HMP shunt pathway. The probably university questions would be like definition of HMP shunt pathway, then its steps, and to mention its significance including the clinical significance. First question, define HMP shunt pathway and mention its significance. So other names of HMP shunt pathway includes pentose phosphate pathway, Dickens-Horecker pathway, shunt pathway, phosphogluconate oxidative pathway. So in other names also it can be asked. So if it is asked for 5 marks questions including the definition, define HMP shunt pathway, you need to first explain what is, you just given a definition of this shunt pathway, what you meant by a shunt pathway, why it is called a shunt pathway. Then you need to explain about the steps, oxidative step and non-oxidative steps. So in detail each step you need to explain, that is by highlighting the enzyme names and what product is formed, what is the end product, starting product. So that flow chart also you need to include. So each step you explain in detail. And the next one you have to mention about the regulation. Next, after explaining each steps, next you have to go through the regulation. 2-3 sentences on regulation you need to write, how it is regulated. Next you need to explain about the significance. Depending upon the marks you can explain the significance. If it is having a higher weightage, you need to explain each significance. So if it is for just for 2 marks, you can just mention the headings, how the significant headings only you need to mention. If it is more than coming around more than 2 marks or 2 marks or something like that, you need to explain every significance. Okay, we'll go to the topic. So first HMP shunt pathway definition you can give. In glycolysis there are few bisphosphate intermediates, but in this pathway they are bypassed, or there is only monophosphate which has been formed. That's why it is called as HMP pathway. And since the reactions is involving the intermediate formation of pentose phosphate, and it is also called as pentose phosphate pathway. So first you need to explain about the oxidative phases. That is step 1. That is glucose 6 phosphate is oxidized by NADP dependent glucose 6 phosphate dehydrogenase to form 6-phosphogluconate lactone. That is the first step, and it is a rate limiting step, and we require NADPH. NADPH, 1 molecule of NADPH is formed in this reaction. Next step is lactone is hydrolyzed to gluconolactone by hydroxylase enzyme to form 6-phosphogluconic acid. That is the second step. And third step is NADPH is again generated with the help of dehydrogenase enzyme. Oxidative step coupled with decarboxylation. The enzyme is 6-phosphogluconate dehydrogenase. 6-phosphogluconic acid is hydrolyzed, dehydrogenated to 3-keto 6-phosphogluconate. It is a transient compound and it spontaneously undergoes decarboxylation to form ribose 5-phosphate. And in this step, NADPH is generated. Glucose 6 phosphate to 6-phosphogluconolactone, then that to 6-phosphogluconic acid, then to ribose 5-phosphate. Next would be the non-oxidative phases. First step is the first step of HMP shunt, isomerization. Ribose 5-phosphate isomerized to ribose 5-phosphate or epimerized to xylose 5-phosphate. Then fifth step is trans-Catalase reaction. Trans-Catalase is a thiamine dependent enzyme, TPP dependent enzyme. It transfers 2 carbon atom from xylose 5-phosphate to ribose 5-phosphate, xylose phosphate to ribose 5-phosphate to form 7 carbon sugar, and pseudo-heptulose 7-phosphate and glyceraldehyde 3-phosphate is also formed. Trans-Catalase enzyme will transfer the group from a donor ketose to an aldose acceptor, and in case of any thiamine deficiency, this trans-Catalase activity would be reduced because it is trans-Catalase is thiamine dependent enzyme. Next step is trans-Aldolase reaction, that is the next group transfer. The reaction involves the transfer of 3 carbon atoms from pseudo-heptulose 7-phosphate to glyceraldehyde 3-phosphate to form fructose 6-phosphate. The next step is trans-Catalase reaction, second trans-Catalase reactions. The another trans-Catalase reactions, 2 carbon atom is transferred from xylulose 5-phosphate to erythro 4-phosphate to form fructose 6-phosphate and glyceraldehyde 3-phosphate. And next step is regeneration of glucose 6-phosphate. 2 molecules of glyceraldehyde 3-phosphate formed in the step 7. It is condensing to form 1 fructose 6-phosphate, and this fructose 6-phosphate is converted to glucose 6-phosphate. These are all the reversal steps which you see in glycolysis. Xylose to ribose 5-phosphate, that is with the help of phosphopentose epimerase enzyme. Then isomerase enzyme is converting to ribose, then ribose 5-phosphate with the help of trans-Catalase, which is TPP dependent enzyme, it is getting converted into glyceraldehyde 3-phosphate and pseudo-heptulose and xylose 5-phosphate. Okay. So all these steps you need to explain in detail, including with highlighting the enzymes. Next you need to write about the regulation of HMP shunt pathway. The first step is the rate limiting enzyme, where it is catalyzed by GPD. The pathway is mainly regulated by the level of NADP. The first reaction catalyzed by GPD is a rate limiting step and is inhibited with the presence of NADPH. The oxidative phase is therefore controlled by the level of NADP, and non-oxidative phase is regulated by the pentose availability. Insulin will induce GPD and therefore will increase the overall pathway. When more NADPH is needed, the pathway proceeds to completion with equivalent of 1 molecule of glucose being completely oxidized to carbon dioxide. Next you need to mention about the significance of HMP shunt pathway. So when you're explaining the significance, you have to divide it into 2 parts, that is the metabolic importance and then the clinical importance. So under the metabolic importance, you need to mention that it is helping in the production of NADPH and pentose phosphate. And this NADPH, the role of NADPH is need to be mentioned, because NADPH is required for various other activities, and this is the HMP is helping in the production of NADPH. So NADPH is required for the reductive biosynthesis of fatty acids, cholesterol, steroid hormones. Then NADPH is also required for free radical scavenging. You all know what is free radical, how it is formed, and how it is affecting the cells, how it is destroying the cells, and how important it is to be. It has to be removed from the cells in order to protect the cells. So NADPH is helping in the free radical scavenging. Then to maintain the RBC membrane integrity. Next, it is required for methemoglobin, prevention of formation of methemoglobin. Next, it is required for detoxification by hydroxylation. It is with the help of NADPH. Then in order to maintain the transparency of lungs, then bacterial activity of macrophages. For all this, it is required, NADPH is required, and this NADPH is produced from our shunt pathway. Next is the clinical importance, that is ribose 5-phosphate is required for nucleic acid activity, then glucose 6-phosphate dehydrogenase deficiency, then drug induced hemolytic anemia, methemoglobinemia, and thiamine deficiency leads to trans-Catalase, reduced trans-Catalase activity. So, these would be the clinical importance. And if it is asked for more marks, you need to explain in detail. Each steps you need to explain in detail. If it is only for 2 marks, you can just mention these points. Next question, that is mention the significance of hexose monophosphate pathway, that is 2 marks. You can just explain the same thing, that is the metabolic importance. Next one would be the clinical importance. With the headings would be enough, but if it is asked for more than 2 marks or something, you can explain each steps. That is free radical scavenging, the NADPH is produced with the help of shunt pathway, and this NADPH is required for free radical scavenging. These free radicals are continuously produced in all cells, and these will destroy the DNA, proteins, fatty acids and all other biomolecules, and in turn that will destroy the cells. So, these free radicals are important that it should be removed from the cells in order to protect the cells from its lysis. So, the free radicals are inactivated by enzymes systems containing superoxide dismutase, peroxidase and glutathione reductase, and reduced GR is regenerated with the help of NADPH. Next function is in RBCs in order to maintain its membrane integrity. NADPH is required by the RBC to keep the glutathione in the reduced state, and this in reduced glutathione is will detoxify the peroxidase and free radicals formed within the RBC. So, NADPH, glutathione and glutathione reductase together is helping in preserving the integrity of RBC membranes. Next function is the prevention of formation of methemoglobin. NADPH is the one which is helping the hemoglobin to keep the iron in the reduced form, that is in the ferrous state. So, it is preventing the accumulation of methemoglobin, and hemoglobin, it is functionless, it cannot carry oxygen. So, it has to be prevented. Methemoglobin accumulation has to be prevented for the proper transportation of oxygen throughout the body. Next one is detoxification of drugs. Most of the drugs and other foreign substances are detoxified by the liver, microsomal P450, which is NADPH dependent enzyme. Next one is in the lengths of ice. In lengths of ice, we have maximum concentration of NADPH, and that is helping in preserving the transparency of lens. Next one is the macrophage bacterial activity. That is NADPH is required for the production of reactive oxygen species and superoxide anion radicals by macrophage to kill the bacteria. Next one is availability of ribose. Ribose and deoxyribose are required for the DNA RNA synthesis, and this ribose is also necessary for the nucleotide coenzymes. The reversal of non-oxidative phases present in all tissues by which ribose could be made available. And in the HMP shunt pathway, one of the main significance is that ATP is neither utilized nor it is produced. So, during the shunt pathway, energy is not utilized. Cells do not use shunt pathway for energy production. Next, you need to explain about the clinical significance of the shunt pathway. GPD deficiency, that is the enzyme glucose 6-phosphate dehydrogenase may be deficient in some persons, which is usually seen during clinical practice, and it is the X-linked recessive trait, and it will lead to drug induced hemolytic anemia. Deficiency is manifested only when exposed to certain drugs or toxins. For example, when we are taking anti-malarial drugs, it stimulates the peroxide formation inside the RBC, which lead to the lysis of the RBC, and GPD deficient cell, the level of NADPH would be very low, and hence further production of peroxide will lead to the cell lysis, RBC lysis. And next condition, that is ingestion of toxic glycosidase, which is present in flava beans, will also lead to cell RBC lysis, flavism. Then during sulfur drugs also hemolysis will be increased and will lead to jaundice and anemia. Then the geographical distribution of GPD deficiency correlates well with the malarial endemicity. Next is methemoglobinemia. GPD deficient persons will show increased methemoglobin circulation, Then during thiamine deficiency, if there is thiamine is required for trans-ketolase activity. So whenever there is a thiamine deficiency, trans-ketolase activity reactions will be lower. Trans-ketolase reaction is measured in RBCs as an index of thiamine status of an individual. The occurrence and manifestation of Wernicke-Korsakoff syndrome, which is seen in alcoholics, and those with thiamine deficiency is due to the genetic defect of the enzyme trans-ketolase. So all this clinical significance, if it is asked for more marks, clinical significance also you need to explain in detail. And this will also be asked as your clinical charts, sulfur drugs and this hemolytic anemia, drug induced hemolytic anemia. Okay, moving on to the next question, that is the clinical significance of trans-ketolase. Already discussed, that is trans-ketolase reaction is a measure of RBC and index of thiamine status of an individual. So whenever there is a thiamine deficiency, trans-ketolase activity will also be lowered.